what is the relationship between the drug concentration and effect. And these are not things that you will completely understand, but it's more to help you be familiar with terminology and an understanding of why, particularly in phase 1 studies, there is a lot more testing that goes on, a lot more blood testing, appointments, vital sign assessments, and things that occur really early on to look at drug distribution and side effects. So these tests then help us evaluate the dose and the schedule. Is more drug better? Does it matter if drug is taken with food or on an empty stomach? And so you'll know that some of the medications that have been approved for kidney cancer must be taken 1 hour before or 2 hours after food, and that is because food in the stomach will change the absorption of the drug. Other drugs, it does not matter, and therefore you can take them irrespective of food intake or meals. The PK and PD testing also evaluate potential drug interactions. And any of you that are on oral therapies, whether they're cancer therapies, blood pressure medicines, Coumadin, which is a blood thinner, you have probably been instructed that there are certain medications or foods that you need to avoid because of this liver system called the cytochrome P450 metabolism. That is a group of enzymes that are involved in drug metabolism. And changes in this enzyme activity can affect the metabolism and the clearance of drugs, including cancer therapies. There are certain medications that are referred to as inducers and therefore increase the rate at which a drug is metabolized. There are also drugs and foods that are inhibitors and therefore decrease the rate at which a drug is metabolized. If your drug is being taken once a day, it may have a 24-hour time frame between dosing. Taking that with an inducer would shorten the life of the drug. So it would take a 24-hour drug and make it essentially an 18-hour drug or a 12-hour drug. Conversely, inhibitors delay or decrease and slow down the rate of drug metabolism, causing a 24-hour drug to become a 36 or 48-hour drug. You then will run into either less effectiveness or worsening side effects because of how medications interact. And so if you're given instructions on avoiding certain medications or foods such as grapefruits, that's why those things are important. And the PK/PD testing helps us evaluate that early on. This is just a graphic of the Sutent, which was originally referred to as SU11248. The patients were given daily doses on the x-axis, or the one across the bottom. There was a 25 milligram dose. You can see a variety of dots, each of which are an individual patient at the 50, 75, 2 patients at 100 milligrams, and 1 patient at 150 milligrams. And at a certain time point, this was the drug level obtained through a blood sample. And so this is a graph that would then show what the variability is in drug concentrations with specific doses. And so that's part of how researchers will correlate dose and side effects to determine what is an appropriate dose and then what is an appropriate dose reduction according to drug levels. So that just gives you a little bit of a feel for it. Let's move on then to phase 2 clinical trials, which really look more specifically at effectiveness while continuing to look at safety and tolerability. Phase 2 clinical trials have a more specific disease group that they're going to be treating, for example, renal cell carcinoma. An individual with breast cancer or colorectal cancer would not be able to participate in a phase 2 trial of a drug in renal cell carcinoma. But there are some drugs that show effectiveness in various diseases in the phase 1 setting, such as Sutent, that showed efficacy in both GIST or GI stromal tumors as well as kidney cancer. So there were phase 2 studies going on concurrently in different places. A phase 2 study of Sutent in GI stromal tumors and phase 2 studies of Sutent in renal cell carcinoma. So only individuals with a specific disease are eligible for a particular clinical trial in the phase 2 setting. And then there are specific clinical settings for each individual phase 2 trial. Drugs typically start in the advanced stage or patients with metastatic disease, and we follow CAT scans for response to determine what the effectiveness is. As these drugs have received FDA approval, many of the drugs in kidney cancer have moved on to clinical trials in the adjuvant setting. In other words, for patients who have had their nephrectomy or partial nephrectomy and who have no evidence radiographically of metastatic disease, will receiving one of these therapies as an adjuvant to surgery for a specified period of time help delay or reduce the risk of having the kidney cancer come back. Along with that type of clinical trial, we are also in the process of doing neoadjuvant clinical trials for patients who have not yet had surgery. Can we improve the safety and the outcome of surgery by giving a drug for a certain period of time preoperatively? So can we give sunitinib or pazopanib prior to surgery, and can patients get through surgery without unacceptable complications? So there's a lot of safety as well as ongoing effectiveness evaluation in neoadjuvant. So those are just some of the settings for which phase 2 clinical trials are done. And as I said before, we continue very carefully to look at the side effect profile, what is acceptable, what is unacceptable in the way of side effects during phase 2 clinical trials, as well as to improve the management of side effects. You know, as we learn what the side effects are, the team of healthcare professionals at a single institution or of multiple institutions can participate in teleconferences. What are we experiencing? What can we do to try to minimize and to try to manage the side effects? So for those of you that are on any of the approved therapies, you can thank the patients that participated in the clinical trials because they helped the healthcare team learn how to better manage and reduce the impact of the side effects. For phase 2 clinical trials, eligibility questions that will need to be met or inclusion criteria include some aspect of treatment history. Clinical trials may say no prior therapy for metastatic disease, or patients must have received one prior VEGF inhibitor therapy. So that would be the criteria looking at effectiveness in sequence. Others will say a particular therapy had to have been given. There may be a time frame since completion of prior therapy. We know in kidney cancer that histology or the type of kidney cancer matters in many situations. So clinical trials may have an inclusion criteria specific to histology. It may be clear cell component, it may be papillary, or it may be broad in that as long as it is histologically or biopsy proven kidney cancer, It doesn't matter whether it's clear cell, chromophobe, or papillary. It all depends on the individual clinical trial. It always is important that we ensure adequate organ function prior to initiation of treatment because, again, we are looking at the safety of the drug and the side effects, and we don't want to put patients at risk of severe side effects as we are learning what organ systems may be impacted by a drug. So we want to make sure that patients are not severely anemic or have significant impairment of liver or kidney function when starting a new therapy. And while these are very specific and absolute for clinical trials, once experience has been gained, while it may have been you had to have a creatinine less than 1.6 to be eligible for a particular trial, once a drug's FDA approved and we know more information about it, then the option becomes, as long as you have reasonable kidney function relative to your physician, that's an option. And again, performance status is also very, very important. What is your functional status and how much impact has your disease had on your functional status, your ability to work and perform activities of daily living? What is the response rate in a particular clinical trial? We're looking at that as far as the effectiveness. And so you will see percentages of a response rate and what is the duration of a particular response. And so this is just an example of some of the data earlier presented on sunitinib based on the phase 2 clinical trials. These are CAT scans, and you can see that this is a CAT scan of the chest, and the yellow arrows on the left point to lymph nodes, enlarged lymph nodes that demonstrate metastatic disease at baseline or the initiation of treatment. And following 8 cycles, you can see that there has been a decrease in the size of the lymph nodes in this particular patient and this particular drug. So this is how we evaluate the response to treatment, is typically using CAT scans. Here again is another one showing a tumor measurement. Again, on the left you see a tumor measurement of 5.1 centimeters, and a little over a year later you see that same tumor is now 2.5 centimeters in length. And so that is how we evaluate the effectiveness, is by doing tumor measurements. We also look at overall, and this is the target study, which was sorafenib versus placebo for patients who had had prior therapy. And what you see on the left is it looks at the response or the tumor change for all patients on the placebo arm and on the sorafenib arm. The x-axis that is a 0 represents the individual patient's extent of disease when they started treatment. Those blue lines that go below demonstrate shrinkage or reduction in tumor volume. The red line that is just shown up is a 30% decrease in tumor volume, which would be a partial response. And the red line above the x-axis, which is at the 20%, shows 20% growth in tumor size. And at the time that these studies were done, that was the cutoff for disease progression. So that is another way in which response to treatment is demonstrated. So phase 3 trials then compare the new versus the standard. And participants have an equal chance of being assigned to either the investigational group or the control group. And there are some studies that have a 2-to-1 randomization, meaning that 2 patients for every 1 will be assigned to the investigational group and 1 patient assigned to the control group. Other studies may allow a crossover, and that allows patients that received the standard or the control treatment to cross over to the investigational treatment if that treatment proves to be more effective. There are some clinical trials that do not allow crossover at the initiation, and they may be amended or modified following data review with the FDA. They may allow that.